This is Crystal Fenn with MedPage Today here in Brussels at the IMPAKT Breast Cancer Conference, where researchers have reported strong evidence for no threshold in the relationship between mortality and the number of circulating tumor cells in patients with metastatic breast cancer. Conference co-chair Dr. Antonio Giordano explained the results at a press briefing here.
This is another attracting tool that we have been investigating in the last 5 to 7 years, and it seems to be very interesting for at least 2 reasons. One is that you can just count circulating tumor cells from a peripheral blood sample, from just a normal blood sample, and apparently this group, that is a joint collaboration from Italy and from the M.D. Anderson Cancer Center in Houston, they are going to present the data this afternoon. They have elaborated a very sophisticated model. I will not go into the details, but this model incorporates also the number of circulating tumor cells that can be detected in a blood sample. And apparently when they put into their sophisticated system also the information related to the number of circulating tumor cells, they can predict very precisely which is going to be the prognosis of metastatic breast cancer patients.
As Dr. Giordano noted, this may have some implications for practice.
Extremely important because as we have been discussing for early breast cancer patients, it's also true for metastatic breast cancer patients that the disease is extremely heterogeneous. We can have patients surviving years and years from the diagnosis of metastatic disease, and unfortunately we have patients who are living just a few months from the diagnosis. So it is quite clear, we know this since many years from the clinical point of view, that the disease is heterogeneous. And now the big hope is that with these new tools, including circulating tumor cells, we can better characterize from a biological point of view metastatic breast cancer, and we can really define which is going to be the duration of the disease and which are going to be the most efficient treatments that have to be given in individual patients. So you have to see circulating tumor cells from one side as a tool that could just predict disease evolution, and on the other side as a tool that could also define the biology of metastatic disease or early disease. And the example that Dr. Picard was giving was exactly the second case. So you use circulating tumor cells to define from a biological point of view the characteristics of the tumor and to treat the tumor based on the biological characteristics of circulating tumor cells.
So circulating tumor cells may have more information to yield in prognosis through use as a continuous rather than dichotomous biomarker. Here in Brussels at IMPAKT, I'm Crystal Fenn, MedPage Today.